reminders, I don't think you need any, but our final lecture exam is a week from today. So it's Monday, 2020. And instead of starting at 7:30, it starts at 8 o'clock. So you get a little bit more sleep or study time or breakfast or travel, whatever it is you need. Goes till 10. Those of you that are in the 10:30 lab exam, the lab section, you're first up for the lab exam this time. And schedule-wise, it's supposed to start at 10:15, but I always give a little bit longer on the lecture exam. So instead of starting at 10:15, we'll start at 10:30. Okay. So that will be overall, obviously, in Humboldt. There are, from the unit, that's digestive and urinary and the reproductive for this week, there are 73 questions. And then from the previous 4 units, there are another 52 questions, give or take. That's where it sits right now, but it won't be more than 130. And as I've indicated in the past, where possible, the questions from the previous 4 units, I will relate back to the material that we've been covering. So in the first unit, for example, we talked about anatomical terminology. So questions relating proximal and distal, what part of the digestive tract is proximal, what's distal, planes of section of the kidney or the stomach or something like that, our longitudinal, sagittal, frontal planes of section, cells. So we've talked about the liver functions in making proteins, about the detoxification. So we have both smooth ER, ribosomal ER, and smooth ER. The organs we're talking about today and Wednesday make steroid hormones. Those are smooth ER. The proximal cells of the kidney do a lot of moving the sodium ions, so they have a lot of mitochondria. So that type of component on the cell organelle. And then for bone, there will certainly be some questions on bones of the extremities. Those have nothing to do with the stomach or reproductive system. All right, but those questions will be, obviously any question is not going to be, what bone articulates or something. It'll be a question of a process that would be important for muscle attachment. Or I won't be saying which of the following bones, carpal bones, is, the most medial or the distal type of thing. It'll be a basic review type question. Bones of the skull. So we've talked about digestion. So there'll be an increased emphasis on bones that have to do with mastication and the nasal cavity and the oral cavity. We talked about cranial nerves. So openings that the cranial nerves pass through, especially those that have to do with something that we've talked about since in respiration or speech or something of that nature. When we get to the muscles, I've emphasized about the respiratory system. I talked about the muscles that support the kidneys. All right, so where possible, accessory respiratory muscles of respiration, the muscles quadratus lumborum and psoas major. There will be some questions of muscles of the extremities, but focusing on what's the function of characteristic functions of muscles of the posterior compartment of the thigh, or as a group. So there are not specific questions on which of the following is the origin of the biceps brachii type of thing, all right? But functions by compartments and by major groups. Same with innervation, unless it was like the diaphragm where we talked about the phrenic nerve. Again, so reflecting back on the areas that we've covered, trying to focus on those, but there will be a few questions on the extremities for muscles, nerves, or something like that where we haven't had a return to the focus. Blood vessels. We've talked about the digestive system, we've talked about the urinary system, blood vessels, obviously, portal vein, mesenteric arteries and vessels, those structures that have to do with that. All right, so that kind of gives you a focus. So there's about 13, with the 4 units, there's about 13 to 14 questions per unit. Okay, and then I kind of looked at, again, those generalities and making that assignment. Your lab exam, see the schedule. And this is a regular exam. So in the past, not for the last couple of years, it used to be a comprehensive exam as well with 54 questions of regular unit exam and 54 questions that were a review. And this is just a regular lab exam. Alrighty, I've had one person request to move from the 10:30 lab to a Wednesday lab exam, which is fine. It's a little difficult to move into the 10:30 because we're already over the number of stations we have. But if you want to move to the Monday afternoon lab exam with Dr. Lancaster, just let him know. He should have enough room if there's 3 or 4 people that want to do that. I think one person is doing that. Otherwise, just check with me if you want to adjust because of travel schedules or because of other exams. Alrighty, so we're ready to work on the reproductive system. I'm kind of just put a sequencing diagram of it over here, and we're going to look at it in more detail. When we compare male reproductive to female reproductive, it has more things to learn about it in the sense that there's more of a pathway. With female, we have the oviduct, the uterus, and the vagina, and that's it as far as structures that the egg passes through. In the male, there's a lot more glands that are involved and more names of the parts of the pathways. In physiology, females make up for it with all the hormones and the endocrine regulation of the menstrual cycle. So when we look at the gonads and their development, for the male, of course, the gonads are identified as the testis, or testes. And for both male or female, embryologically, these develop up from mesoderm, that same tissue that forms connective tissue, and the liver, organs, skeletal muscle, smooth muscle. And so these structures, and this is for free, right, this diagram right here, but on the posterior wall of the embryo is a mass of mesodermal tissue here that develops into both the kidneys and the gonads. There's several terms on your lecture notes, but the main ones that I'm interested in is, I'll underline and write on the board in just a moment. The kidneys, and I did mention this when we were talking about the kidneys, but the kidneys are unique in their development in that 3 separate kidneys appear. Only the last 2 are functional. They'll appear and disappear, and then another kidney will develop and disappear, and then finally we have our mature 3rd kidney. We don't see that with any other organ development in the body. The first kidney doesn't function to filter. Its function seems to be to induce, or its action seems to be to induce or to trigger the development of the gonads and the secondary kidneys. And so then the second kidney has a long duct, and that duct is known as the mesonephric duct. That second kidney is called the mesonephros. For males, it is the mesonephric duct that is converted or further differentiates into male reproductive structures. All right, not the gonads, but this will become the efferent ductules, it will become the epididymis, and it will become vas deferens, and even the upper part of the urethra. The vas deferens is also known as ductus deferens. So this embryonic structure, as we'll see on Wednesday, or talk about briefly on Wednesday, this disappears in the female. So this is the male developmental structure, and in the female, the mesonephric disappears, and another duct, the paramesonephric duct, remains. This has to do with the Y chromosome and the secretion of secondary sexual nephrons, sexual hormones. And so this just appears in the female. In the female, the paramesonephric duct remains, and this becomes the oviduct, uterus, and the upper part of the vagina. So this is under gonadal influence. In fact, there are some individuals, there's an island in the Caribbean that, because it's an island, there's a lot of interbreeding, and that's affected some mutations of the genes. And so about, a small percentage, and I'm just thinking off the top of my head here, but it's less than 20% of males when they are born have insufficient enzyme to convert testosterone to the hormone that is called DHT, dihydrotestosterone, 2,4-dihydrotestosterone. And without insufficient enzyme to convert to that hormone, the mesonephric duct does not develop completely, and the paramesonephric duct remains. So these children are born with vaginas, but they don't have gonads. They have testes, but the testes are undescended. And when they reach puberty, there's a flood of testosterone, and at that time sufficient secondary hormones are converted from testosterone to DHT, and that's when their clitoris and the labia majora structures are mostly homologous in the female finish forming a penis, and the girl children turn into boy children at around 10 or 11 years of age. They're used to it because that's what happens. So I guess, is it a girl or a boy? It's well, we'll know in 10 years kind of a thing. But without going into all the detailed embryology, I just want you to recognize that of these embryonic structures, the mesonephric duct forms the male structures under influence of hormones, and paramesonephric duct forms the female structures. And if it is a male but without sufficient hormones, the persistence is as female secondary characteristics. Okay. In fact, the video that I saw on this was called "The Fight to Be Male." What happens if the male hormone is not present? All right, so as I indicated, up high near the kidneys is where we see the gonads developing. And in the female, these testes move only into the pelvis. In the male, they move down the posterior abdominal wall behind the peritoneum and are pulled down into the scrotal sac. So in both male and female, there is a structure attached to the gonad. One is an upper gubernaculum, and the other is a lower gubernaculum. It's just a band of connective tissue, and in the male, this lower gubernaculum is attached to the scrotum, to the scrotal sac. And as it shortens, it pulls the testes down behind the peritoneum, along the posterior abdominal wall, through an arching layer of muscles known as the inguinal canal, and into the scrotal sac. Right. So finally, this usually happens about 7 months of fetal age, so about 2 months before the male fetus is born. And so we end up, the upper gubernaculum disappears, and we end up with the lower gubernaculum anchoring it to the scrotal sac. And so this is known as the gubernaculum testis, or just called gubernaculum for short. Some infants are born without the testes descending, which is okay as long as they descend before puberty is reached, because at that time, with the higher temperatures in the abdominal wall, it's not conducive to sperm development, and the male will become infertile. Okay, but this can be done surgically before that time. These same gubernacula occur in the female. All right, so I'm going to start a chart over here, and we'll continue it on Wednesday of what are homologous structures. Homologous means equivalent to. All right, so in the male versus in the female. So we have our mesonephric duct in the male and our paramesonephric duct in the female. In the male, we have the upper gubernaculum, which disappears. In the female, it's going to become the ovarian ligament, and we'll talk about this on Wednesday. All right. The lower gubernaculum notice how it passes through the inguinal canal and anchors the testis to the bottom of the scrotal sac. In the female, it passes through the inguinal canal and it attaches within the large fleshy lips on either side of the opening of the vagina that we'll talk about on Wednesday, the ones that have hair follicles on them, called the labia majora. And so that structure in the female is known as the round ligament. Not the same as the ligamentum teres, a different round ligament. And that anchors the uterus facing forward. So before we talk about the testis itself, I want to introduce you to the concept of the inguinal canal. And Netters has a diagram that is quite useful. I'm going to show it to you here. We're not going to go through all the layers, but there's a couple that I want you have to identify for lab. I want to illustrate it here. So this is looking at an anterior view of the pelvis. And you'll notice there's I don't know how many of you played with wooden building blocks when you were kids, but some of the blocks are cylinders and some are just blocks, but some of them are arches, wooden arches. And so imagine each of these layers as a wooden arch that's slightly offset. So the more deep arch is more lateral, and as we move superficial through the abdominal wall, each anterior subsequent arch becomes more medial. So we start here at the front, and this structure is called the spermatic cord. This is a channel that I'm going to trace with the light. This channel is what the lower gubernaculum, pulling the testes, passes through to pull the testes down into the scrotal sac. And following with it are testicular vessels, testicular arteries and veins, and those are still found in the canal. And included in that then is the duct, the vas deferens, that the sperm has to pass through going in this direction to pass posterior and behind the bladder. So these series of arches at the front, this is the external abdominal oblique, right here. No muscle fibers, but the fascia from that muscle comes down and forms the roof of the inguinal canal and folds under and forms the floor as well. So this opening right here, called the superficial inguinal ring, is formed from fibers of the external abdominal oblique. As we move posterior to that, deeper to that, the next layer is our internal abdominal oblique, and that actually gives off some skeletal muscle fibers. These skeletal muscle fibers, if I could get a good view here, these skeletal muscle fibers pass down into the scrotal sac. This is a model. So this would be the scrotum here. And these skeletal muscle fibers are seen along right here. And if I can get a better, here we go. So this is a deeper cut. Here again is the superficial inguinal ring. And here are these skeletal muscle fibers of the internal abdominal oblique. At this point, they're called cremaster muscles. Okay, they're voluntary. But there's a cremaster reflex stroking the side of the thigh that causes muscles to contract and pulls the whole scrotum up closer to the body and this increases the temperature of the scrotum. Then as we go and look at these arches, the next layer is the transverse abdominis, and that forms an arch, and then we have the fascia, and finally the peritoneum. Okay? So as these form arches, the deep inguinal ring is, of course, deep. It's on the interior surface of the abdominal wall, and it's lateral to the superficial inguinal ring. So the superficial inguinal ring is superficial and medial. The deep inguinal ring is lateral, obviously, and deep. Now, a friend of mine, a colleague, he's doing research here for his master's, but he also is one of our teaching IA's instructional assistants at AR. He just had a double inguinal hernia repair last week. A hernia is when there's a weakness in the abdominal wall or in any muscle wall. You can have an esophageal hernia, where the esophagus passes a hiatal hernia, where when the esophagus passes through the diaphragm, that becomes weakened, and part of the intestines or stomach can bulge up into the lung tissue. You can have an umbilical hernia, so a newborn baby crying a lot can stretch that opening, and intestines can bulge out into the area where the umbilical cord is. So in an inguinal hernia, there are two types. The weakness in the abdominal wall can allow a loop of intestine to pass straight through and enter the inguinal canal here, right? Passing not through the opening, but straight through. That's a direct inguinal hernia, going the direct route. An indirect, the loop of intestine actually passes through the deep inguinal ring, but both of them enter the inguinal canal. And the problem with that is there can be a twisting of the intestinal loop and a blockage of blood flow, and so those are typically repaired. So looking on the deep surface, this is the breastplate, if you will, so cutting off the anterior abdominal wall here and lifting it out, or climbing inside the abdomen and looking forward. You're looking at the rectus abdominis here, and the transverse abdominis muscle here. Here would be the external iliac, and here's the vas deferens coming out of the inguinal canal and going towards the bladder, and here would be the testicular artery and vein. So you're looking at the deep inguinal ring, or in your lab manual it's called the external inguinal orifice, from the inside surface. But the concept is it's this angled passageway. If there is a hernia, all right, so here's a direct inguinal hernia, meaning the most direct route is straight through all of these layers of muscle tissue. The indirect is coming in through the deep inguinal ring. Now it seems backwards, but let's say that that's the only opening out of this room, okay? And so to go out of the room, I have to walk around all the chairs and go out that door. That's a lot longer pathway than to just walk through the wall. Let's say this wall was to the outside. If I just went right through that wall, that would be the most direct pathway. Maybe not the easiest, but the most direct. All right, so direct inguinal ring is straight through the abdominal wall in this direction, like this, whereas indirect is coming through the deep inguinal ring. All right, so let me just write some of those structures up on the board, but before we leave that, if we open up the premasteric muscle, then we see the contents of the inguinal canal. And those include the blood supply, nerves, and the vas deferens itself, which is carrying the sperm away from the testes. Females have an inguinal canal, but because the only structure that passes through it is that round ligament, they seldom develop an inguinal hernia. So I'm not going to draw all the components that were on, that might even be in your lecture notes, but just the ones that you need to know. So here will be inguinal canal, the scrotal sac. And we'll put the testis. the reason we have all these layers, of course, is as the testis passes down, it's pulling these layers of the wall through it, with it. Just deep to the skin, we have a layer of smooth muscle. It's within the connective tissue, deep to the skin. This is smooth muscle, and so it's going to be controlled by the autonomic nervous system, and it's named the dartos muscle. It will contract and reflex to cold. So there's a famous Seinfeld episode where George is coming in from the ocean and he's, embarrassed by what's happening to his scrotum, and he's explaining it away as the water was cold, contracts and pulls up close to the body wall. And so that's a part of the autonomic response of the smooth muscle. Then we have the layer of the external abdominal oblique, the fascia, which I'm not going to label because you don't need to know it, but this would be the fascia of the external abdominal oblique. Fascia is just a sheet of connective tissue. And then we would have the layer of skeletal muscle fibers. And this is the cremaster muscle. So I want you to know that that's the cremaster muscle. I also want you to know that it's derived from the internal abdominal oblique. It's skeletal. So now we've got three muscles in this unit that has specific names. The bladder of the smooth muscle of the bladder. Anybody remember what that one's called? You might have to see that on your quiz today. Detrusor. Okay. We have the smooth muscle of the scrotum, which is dartos. Those both start with D. And then we have the skeletal muscle of the scrotum, which is the cremaster. Okay. Now, because remember the testes are retroperitoneal as they develop in the posterior abdominal wall. So as they're sliding, being pulled down the posterior abdominal wall, they pull all these layers of the abdominal wall with them. They grab, well, not with fingers, but they grab a hold of the peritoneum as they're kind of sliding down and pull the peritoneum down into the abdominal wall with them. All right? They don't pass through it. They're still behind it. So this last layer that I'm putting in here is peritoneum. But notice it's only on the front of the testes. And this is, you guys didn't know that you actually have two vaginas. It's called tunica vaginalis. And it's derived from peritoneum. It's typically collapsed. So it's open only to the abdominal cavity, but sometimes it will fill with fluid and cause one of the testes to become enlarged, and it's known as a water sac or hydrocele. You've heard of that. They just withdraw the extra fluid via syringe. Sometimes they need surgery to close the cyst-like structure. Okay? So this does not go completely around the testes like the other layers do. It's just found on the front of it. So in the spermatic cord then, and these are just the walls of the spermatic cord. It's going to contain the vas deferens, which we'll talk about in a few moments. This is a hollow tube through which the sperm travel to reach the urethra. It's going to contain blood vessels, so our gonadal artery, the testicular artery. And then I don't know if you noticed in that previous diagram of the model, but instead of a single testicular vein, it forms a network of veins. And so this is known as the pampiniform plexus. So here would be the testicular artery, and then the pampiniform plexus is a network rather than a single vessel of veins that wrap around the testicular artery. The purpose for this is to create a countercurrent exchange. So these two have a countercurrent exchange of heat. Temperature regulation is very important for the testes. And so if the arterial blood is going directly into the testes, that would carry too much energy, heat energy with it, and would be harmful to the development of the sperm. And so the veins wrap intimately around it, and heat is transferred from the testicular artery to the pampiniform plexus. And that warms up the testicular artery, warms up the pampiniform plexus as that blood leaves the scrotal sac. But then it's coming in contact with blood that's just left the abdominal cavity. So that blood is even warmer, so there continues to be an exchange. This actually occurs in the flukes of whales that are in the Arctic Ocean. They're mammals. They use energy to create heat, and they would have to use up a lot more energy because their large tail surfaces exposed to the cold Arctic Ocean will lose a lot of heat. So as blood flows out to the tails, it has a very similar countercurrent flow with the icy venous blood coming back in, and conserves that heat, warms up the venous blood as it comes back into the abdominal cavity, so the whale doesn't have to use additional energy to do that. And that's what we see here, is an exchange of heat between the testicular artery and pampiniform plexus. So in lab, if you identify this as the testicular vein, that is not incorrect. It is a form of the testicular vein, but for full credit, pampiniform plexus, if it's wrapped around the structure of the testicular artery in the spermatic cord. Then we have nerves. We have the genital femoral nerve, which is part of the lumbar plexus. Okay, so those are the major components of our testicular spermatic cord. All right, so now let's move to the testis and look at the structure of the testis in more detail. There's a bit more of this in the online lecture notes, but what I'm putting on the board is what I want you to focus on, all right? Because these are notes from my class at AR, and we have a little bit more lecture time available than you guys do. So what I'm covering on the board will be what you're responsible for. All right, so can I erase the diagram of the scrotal wall? And we'll look at the structure of the testis. All right, the testis is a large oval, well, about the size of an egg, but I'm going to draw it large. Oval structure, it's covered in a connective tissue capsule, known as the white capsule or tunica albuginea. That's a dense, irregular connective tissue. Fibers from that capsule divide the testes into lobules. So this would be like if you peel an orange, it comes off in segments, because the inner aspect of the peel of the orange is dividing the orange into segments. It's similar to that fashion. Within the lobules, we have about 15 to 20 lobules, seminiferous tubules within each lobule. I'm just going to draw one. These tubules are highly coiled, much more than I'm actually drawing. And then they come together and join kind of in the center here of the testis. And these tubules are identified as the seminiferous tubules. Okay, we'll come back and talk about them in a little bit more detail. But the straight section of two seminiferous tubules joins together, and that's what it's called. The tubuli recti, or most commonly referred to in English as the straight tubules. Okay? Go a little bit farther over here. Straight tubule, I believe, is in your lab manual, but you might see that terminology. And then there's a network here in the center where they form several channels. And this network is known as the Reedy testes. And then finally, remember I said that the mesonephric duct forms efferent ductules. So we start to see that structure now from the mesonephric duct. I'm drawing it in a different color because it has its origin from the mesonephric duct. So efferent ductules or ducts. And then these join together to form a single structure which is highly coiled. Just one, although it's very highly coiled. And it's found on the posterior aspect of the testis. And this is identified as, like trying to spell Mississippi. A whole bunch of I's and a Y. That's the epididymis. Okay, a single coiled structure. This region is known as the head. Then we have the body, and finally the tail. And it is the tail that will then form the vas or ductus deferens. Structure that passes through the spermatic cord and carries the sperm towards the urethra. So we're going to take a closer look at the seminiferous tubules. The sequence that the sperm would travel is from the lumen of the seminiferous tubules into a straight tubule, into the Reedy testes, into the efferent ductual, head, body, and tail of the epididymis. And then they stay there until emission occurs, all right, until emission is just prior to ejaculation. And that's the events that will occur in their maturation. Now, the seminiferous tubule, or tubules, are hollow tubules formed of a simple columnar epithelium. So if I were to draw that just as a small cross-section, we would have, and it's very difficult to actually see these cells that I'm drawing. These are simple columnar cells that support the development of the sperm. And as such, they are identified as, the older name is Sertoli cell, but they are also known as sustentacular cell. And I don't know if you recall the myelination of axons. Remember how the Schwann cell or the oligodendrocytes wrap tightly around the axons? But even if an axon was unmyelinated, here would be the Schwann cell. Remember how the Schwann cell would still intimately be associated with the axon. And it looked like the axon was actually inside the Schwann cell, but it was really just the plasma membrane wrapped around it. That's what happens with the seminiferous tubules and our developing spermatocytes. You don't need to know all the DNA changes that occur, all right, but I'm going to take you through the stages. So I'm going to draw a couple of these sustentacular or Sertoli cells, quite large. But first of all, I want to diagram the developing sperm cells as they pass through. So we have a layer of basement membrane, like we do with every epithelial tissue. This helps to protect the developing sperm cells from the male immune system because some of the proteins they have on them are foreign. And along the base of this basement membrane, the base of the cell, we have gonadal cells that have not yet started to mature. So these are the least differentiated, and they are identified as spermatogonia. Or spermatogonia for plural. Then they will undergo cell division, and they will divide, leaving one behind, and then the other one will start to migrate towards the lumen. So this will be the lumen of our seminiferous tubule. So one that gets left behind, okay, this one got left behind, and then the next one is going to start to undergo meiosis. It's going to undergo the replication of all of its chromosomes in preparation for cell division. Those chromosomes become condensed. So we have duplicated chromosomes. We can actually visualize them condensed within the cell structure. And although you don't need to identify these specifically, I'm just going to go ahead and label them in case you're familiar with them. These are known as primary spermatocytes. In lab, you just need to know that they're developing spermatocytes. Okay, you don't have to identify, and even for lecture, you don't have to be able to identify these different stages. Then this cell undergoes meiosis. It actually divides. This one is doing the same thing over here. This cell divides, but a small cytoplasmic bridge remains connecting the two. The chromosomes separate in a meiotic form, so we don't have a full complement. We're reducing the number of chromosomes because the entire intent, when this is a mature sperm, it's going to fuse with an egg, and each one has to have half of the amount of chromosomes. Physiology class, but these are now identified as secondary spermatocytes. Notice I'm drawing just the spermatocytes. I'm not yet have put in a sustentacular cell. These divide again. The nuclei are smaller, but they're still connected. Cells are still connected via a cytoplasmic bridge. The nuclei are very condensed because they're not going to be producing hormones, cellular products anymore. And then finally, they start to change their shape. They start to actually look like sperm as they lose cytoplasm, because this is way too much cytoplasm to be transported. The egg is going to have the organelles that the new cell needs for further synthesis and division. And so this is streamlined, all right? I don't know how big the scrotal sac would be if this didn't happen, but something like this, okay, rather than the smaller structure. We just lose all this cytoplasm. So now these cells start to look like sperm. Again, they change from heads to tails. There might still be some slight cytoplasmic association. So here's the relationship then of the sustentacular cell. it looks like these cells are actually within the sustentacular cell when they are not. We can't see these fine divisions. So what I've drawn here would be one sustentacular cell, here's another one, here's another one. So they have a lot of folding of the plasma membrane as they wrap around the developing spermatocytes. Their nuclei are distinct, very angular, and a distinctive nucleolus. Okay? So these are the developing spermatocytes. These are now known as early spermatids. These are known as late spermatids. And it's not until they're actually freed within the lumen of the spermatozoa, the seminiferous tubule, no longer associated with the cell, that they're known as spermatozoa. So the sustentacular cell secretes some proteins that bind testosterone that come from other cells. I'll diagram them in just a minute. Keeps it in high concentration because that testosterone is necessary in the development of the sperm. So if we look at the seminiferous tubule in cross-section, so look at the testes in cross-section, because the seminiferous tubules are so highly coiled, we see numerous, kind of like the proximal convoluted tubule and distal convoluted tubule, we see numerous cross-sections of the seminiferous tubule. And then in between them, where there's lots of blood vessels and capillaries, we see cells known as interstitial cells. The old name used to be the interstitial cells of Leydig. And these cells synthesize the male hormone testosterone. It goes into the bloodstream because they are endocrine, but because the Sertoli cells make a protein that holds on to testosterone, they are held in high concentration around the Sertoli cells. Again, we'll come back to this on Wednesday to relate these structures to you. The interstitial cells of the male are homologous to cells, a structure in the female that we call the theca interna. Both of these cell types secrete testosterone. Our ovary cells make testosterone. The Sertoli cells are homologous to the cells directly around the egg, known as follicular cells. And both of these cells take that testosterone and convert it to estrogen. So, of course, this happens a lot more in the female. We have a lot more estrogen than we have testosterone. But if a woman has cervical cancer or very large fibroid tumors or for some reason needs to have her uterus removed, and they do a radical hysterectomy, which includes removal of the ovaries, oftentimes she's given testosterone as a supplement. Because without the testosterone that her ovaries produce, there's a radical drop in her sex drive. And so she may be given supplemental testosterone to replace what her ovaries are not making. All right, so we'll come back and talk about these cells on Wednesday with the female, but I wanted to give you the equivalent structure in the male. So now the sperm move through and into the lumen as spermatozoa. I'm limiting myself here on diagrams. So let's look at the structure of a sperm. So this is a spermatozoan. This is the sperm that is no longer associated directly with the membrane of the sustentacular cell. It's streamlined, if you will. It has a different shape depending on what species it's found in. In a pig, the head is shaped like a ping pong paddle. In a hamster or a guinea pig, the head is shaped like a sickle. So this is known as the head. This is the midpiece. And this is the tail. Within the head, one of the Golgi sacs, we're no longer making proteins, but one of the Golgi cisternae remains and forms a cap over the nucleus. Kind of like a stocking cap that you might pull down over your head. So it looks U-shaped here, but it's actually completely covering. And this is known as the acrosome. From a Golgi sac. And it contains enzymes which help the sperm digest their way through a protein covering around the egg when it's released from the ovary. So without sufficient numbers of sperm, there's insufficient enzyme to create a pathway for the sperm to actually gain access to the egg's membrane and to fertilize it. So that's found in the acrosome. Then within the head itself, we have, of course, the nucleus. And this has just N. So our cells, our somatic cells are 2N. We have 23 chromosomes from our mom and 23 chromosomes from our dad. So this would be just N, which is 23 chromosomes. So that when it fertilizes the egg, it's providing half of the DNA material. In the midpiece, we have a couple of things. We have the origin of the microtubules that are going to be found within the tail, also known as a flagellum. And this is like a very long cilia. So the microtubules are found in doublets. So they slide back and forth just like a cilium. All right. So these are microtubules, and that's essentially what the flagellum is formed of. Remember, microtubules are motile in our cilia. Actin is what we find in our microvilli, and they're non-motile. And then surrounding the microtubule structures that are having their origin in the midpiece, we have rings of mitochondria. And this, of course, is where we're producing the ATP for the movement of the microtubules. The spermatozoa are not yet capable of swimming. Okay, the enzymes that allow the tails to beat are not mature. The acrosome is not capable of, the cell is not capable of undergoing an acrosome reaction. That occurs when the cell is in the female reproductive tract, and this part disappears and releases those enzymes around the egg. So all of that can't happen. If we were to take a spermatozoan from the lumen of a seminiferous tubule and place it in a petri dish with an egg and the appropriate fluids, it would just lie there. 20, 50 million of them would just lie there. Okay? So it's propelled out of the seminiferous tubules through the reedy testes, into the epididymis, and to the tail of the epididymis, primarily by a flow of fluid. So it's kind of just carried along by the current. And it moves to the tail and resides there. And over time, changes occur in the plasma membrane that allow it to undergo this reaction. Enzymes mature that allow the microtubules to slide past each other. So that when it is ejaculated, they are capable of swimming through the female reproductive tract. Okay? So that maturity occurs in the tail of the epididymis. So that brings us then to our next structure, which is the epididymis. So that's what I drew here. So it's a single, long tube that is highly coiled. And again, maturation of the sperm occurred here. The ability to release those enzymes and the ability of the tail to beat, even though it doesn't actually swim out of the epididymis. So this is a columnar epithelium, very long microvilli. They're not cilia, very long microvilli. And then we have the vas deferens. The movement of the sperm, may I erase the diagram of the tail? The movement of the sperm, the movement of the sperm into the vas deferens, along with the movement of fluids from the glands, is a process known as emission. So emission is movement of sperm into the urethra and addition of glandular secretions. Also to the urethra. So that is emission. Okay. Now the seminiferous tubule, the vas deferens is a muscular tube that passes through the spermatic cord, through the inguinal canal, and then passes laterally to the posterior aspect of the bladder. So if we're looking at a posterior view, of the bladder, we're down in the pelvic cavity. We're going to have our ureters coming around because they pass into the bladder posteriorly as well. So here would be the ureters coming into they're kind of passing at an angle through the posterior abdominal, posterior wall of the bladder. And then we have the vas deferens, or ductus deferens, kind of looping over them. Then they dilate to form the ampulla. Just lateral to them are a pair of glands known as the seminal vesicles or the seminal glands. They also are kind of, these are the seminal vesicles. And the two join together. My bladder is a little bit too low here. The two join together to form ejaculatory ducts. So the content of the vas deferens, which is the sperm, and the content of the seminal vesicles, which is a bicarbonate-rich fluid. It has nutrients for the sperm in the form of fructose and citric acid, which the mitochondria and the cell can use directly to form ATP. There's also clotting agents that will coagulate the seminal fluid for a short period of time as the sperm become oriented towards the oviducts, and well, I'm not going to write it on there. It's another protein that breaks down the fibrinogen. Okay? So now these ejaculatory ducts pass through the wall of another gland known as the prostate gland. And the prostate gland is like a donut that completely surrounds the urethra, passes through it. If we were to look at this on a sagittal view, so if I were to drive, this is a sagittal view of the prostate gland, we would have the ducts coming in posteriorly, passing through the prostate gland and opening into them in that fashion. Okay? So the prostate gland has a whole bunch of smaller ducts, multiple ducts, that secrete into the urethra. Prostatic secretions are a little bit more acidic, all right, but they have a lot of zinc, which is an antibiotic, and other chemicals that help to protect the sperm. This first portion of the urethra is identified as the prostatic urethra. And we have three regions of the prostatic urethra. So I'm going to move this screen over and move that whole area up a little bit higher. So this would be the internal sphincter of the bladder. And our external sphincter now is inferior to the prostate gland. This is known as, remember the urogenital diaphragm that we had for, so urogenital diaphragm also known as external urethral sphincter, also known as deep transverse perineal muscle. So this is prostatic urethra. It's lined with transitional epithelium, just like every other part of the urinary tract. This is the membranous urethra, where it passes through the muscle. It's the most narrow. So someone has to be catheterized. That's the most difficult part to catheterize. And the rest of the urethra is found within the penis itself. So this is known as the penile urethra. and it passes through a body of erectile tissue called the corpus spongiosum. So it's also known as the spongy urethra. Now before we look at the structure of the urethra, we have two more glands that are found on the inferior aspect of the urogenital diaphragm. These actually secrete first in emission, and they create a substance that flushes out the urethra of any urine residue and helps to lubricate the tip of the penis. And so these are known as vulvourethral glands. That's a name that they share with the female. The female has vulvourethral glands. Their male-only name is calipers. So either term is appropriate. All right, let's look now at the structure of the penis. And when we do the female, I'll also go over these same structures because we find them in the female. But as sperm moves through into the urethra, remember emission was movement of sperm and secretions into the urethra. Ejaculation is the movement of semen, which is the sperm and its fluid, through the urethra. All right, now, the structure I'm going to show you for the components of the penis, I'm going to diagram the pubic symphysis. Actually, I need to draw this down a little bit lower. Here would be the pubic symphysis, obturator foramen. Okay. The penis is formed of three bodies of erectile tissue. The reason it's called erectile tissue is it's full of arteries, small arterioles. Normally, the sympathetic system is in control of whether vessels constrict or dilate. But in the case of the penis, it's parasympathetic. Reproductive activity is homeostasis. And so under release of acetylcholine, these blood vessels dilate. And blood flows into the penile structures. As they dilate and as blood flows in, that causes the veins to be collapsed as the vessels are expanding, and blood can't leave. So the penis becomes turgid and enlarged, and that's called erection. Okay? So that is specifically the dilation of penile arteries and arterioles. So that erectile tissue is of three parts. If I were to draw a cross-section of the penis, on the dorsal surface, so the penis is not expanded. This is identified as the dorsal surface. Remember a horizontal structure, so it's identified as if it was erect. So that's the dorsal surface. This is the ventral surface. Okay? So in the dorsal surface, we have two bodies of erectile tissue known as the corpora cavernosa. And in the center, always reminds me of a monkey face, sorry guys. This is the corpus spongiosum. And this would be the penile or spongy urethra. Okay? So now if we look at the corpora cavernosa, where it's still attached to bone, so along the rami, this part where it's still attached to the bone is called the crus. Of the penis. Two of them are crura. Where they come off of the bone, now, These two would be the corpora cavernosa. So attached to the bone is the crus. Free of the bone is corpus spongiosum. So this is pointing up towards the abdominal wall, so that would be in a more erect position. Between these two, we have a single structure known as the bulb of the penis. The urethra enters the bulb of the penis. All right, so directly below the urogenital diaphragm, it's going to enter into the bulb of the penis. This is a single structure. So it would be this structure that we see right here that's forming. And it's on the ventral aspect of the penis. So up here, where it's part of the penis, this is identified as the corpus spongiosum. So the crus becomes the corpora cavernosum, and the bulb becomes the corpus spongiosum. The urethra is found within the corpus spongiosum. At the tip of the penis, the corpus spongiosum expands and forms a mushroom-like cap that covers up the tip of the corpora cavernosa. And this is what you know as the gland penis. A very sensitive area, and it contains the opening of the urethra. So where we have the skin covering the structure of the penis, it's folded back on itself and fuses, and that is the foreskin or the prepuce. All right, so this is a retractable covering of skin. And in male circumcision, this is what is removed kind of right here at the base. Okay. Usually done within a few days to a week after birth. I have a babysitter whose son was circumcised at 5, and then there are sometimes adults who are circumcised. We'll talk about how this relates to female circumcision on Wednesday. Okay? Alrighty, so lab, you have a quiz on the urinary system. That's our last required quiz. We'll go over male reproductive structure, and then on Wednesday there's an optional quiz that may replace your lowest quiz score if you choose to take it. That will be over the male reproductive system from today.